Let's talk about species. The species concept is one that we use a lot in biology, very widely used, and yet it's kind of a squishy concept. There are no firm boundaries on what a species can be or what it needs to be. These are taxa of convenience, as are all taxa. And that's, a little upsetting maybe to hear, but if you think about it, a species is going to have a set of rules. So it's going to have a description behind it. The description is going to say, these are the features that you should expect to see in a member of this species. Now, a member of the species may be incomplete. You may define, this is what the full brachiopod looks like, and you might have one that's been damaged. So you might only have one valve of it. But it can still fall within the constraints of the species. And so it would say, well, okay, if these parts are present, this is what you look for. Now, modern species can be separated by DNA, and that's the smartest way to do it. But when we're talking about paleontological species, those are going to be based on morphological characters, which means lumps and bumps and the shape of the thing. Yeah, if it looks like a duck and it's a fossil, then it's a duck. Yeah. And you just got to define what duck-like features you're going to look for, basically. So that's how it works. There's a little more to it. So here, let's jump over here. There has to be, for a modern species, a thing called a type specimen. And the type specimen is like the ideal. I think the ideas for these come from an older time. And there were all sorts of people trying to imagine the perfect human, for example, the perfect bird to put in your museum, something like that. So the type specimen, and there needs to be one of these type specimens for each species, is going to be the individual. And you want to preserve this one. It needs to be in a museum where it is publicly available. You need to publish this so that it can be seen by people who aren't in that particular museum, ideally. These rules are all there so that taxonomy can work as a field, so that other people can figure out, well, how do I have this thing in front of me? What do I call it? What should its name be? Yeah, I'm glad we don't do this with children. Yeah, what should this name be? Let's consult some museums. Yeah, see if we can figure it out. So here we go. This is Carlos Linnaeus, the guy who invented the two-name system for biology, or at least popularized it. Linnaeus has been designated as the type specimen for Homo sapiens. That's you and me. Which means that all of us exist we can only be in the species by comparison to the features that are or were exemplified by Mr. Linnaeus over here. Now, a problem comes in, which you may have spotted already. What if you happen to be female? You would have some different organs and some different appearance from this dude over here. And the answer is yes, you would. And so ideally, a species description should encompass, like, the female looks like this, the male looks like this, but there can still only be one type specimen, and you pick one or the other to be the type specimen. From its earliest days, it would appear that science had no idea what a woman was. I think that's very cute. So this guy, who is now just bones, is the thing to which all humans must be compared, technically. So these rules were put in after a lot of species were named, because it was, I don't want to say the Wild West, because it was wild Europe, Western Europe mostly, at the time, where the new species were being named. They were sending out explorers around the world. These are your Charles Darwins and all the other people competing in that big race. And they were collecting specimens, and they were sometimes taxidermying them to bring them back home to Mother Europe, and so that they could be in a museum, a repository, and they could become the type specimens. Yeah. Some early species, though, don't have a type specimen because it came in before those rules. And it'll be a description sometimes, a little written paragraph, like it should have, 5 arms, 3 legs, and 17 eyes, or whatever it is. And hopefully that should be enough that you can actually identify it, although some of those descriptions are pretty bare bones, and actually you would struggle. You would definitely struggle. For many of the earliest species, when they are published, they either are published only as words, which is tough, or they are published with a line drawing. And not all artists are equally skilled. I'll put that out there. There's some line drawings where they would attempt to fill in missing parts, for example. And so you'll compare this to a complete, recently collected specimen and say, wow, these parts are different. And it's because they weren't there. If you can find the original type specimen in a museum, you can sometimes look and say, oh, yeah, they were making those parts up. So there you go. These are some of the types, and we'll go over some of the rules for how you make types and how you make species. The 2 things you need to make a species are a type specimen and a description. A description is a written set of rules, because taxonomy is all about the rules. And these are the rules for if you fit within this group, or if you're closer to this group than anything else, then you are in this species. These are the rules to get into the club. Yeah. The description serves as a bouncer at the gate of the club, determining which ones get in and which ones do not. All right, types of types. Yeah. They're all referred to as type specimens, but there are different kinds, and they have different ranks. So the king or queen of types is the holotype. This is the one to which all others are related. This is the ideal. And this is the single individual. Yeah, and it has to be just one that will define the species. When you're talking about something like a DNA-based, species designation, then it's much harder to be ambiguous, or at least in modern animals, they tend to be very close to each other, and not that close to anything else. So it's doable, yeah. So the holotype is the single type. It tends to be a complete specimen. Those are the best ones because they have all the parts on them. And usually a mature specimen, like that's a polite word for an old specimen, is better than a young specimen, because the old specimen will often show all of the growth, like full development. Because if your type specimen for a butterfly is a caterpillar, and then you look at a butterfly and you look at a caterpillar, you say, those are totally different things. But you got it at different ages. And in the butterfly, you can actually see some traces of the caterpillar that it used to be, is the idea. I mean, that's an extreme example. And thankfully, caterpillars do not fossilize well. So neither do butterflies, really. Let's see. Yeah, holotype. Second kind of types are paratypes. Paratypes are like the entourage. These are the supporting types. They're going to be all around the holotype. They're going to be keeping the holotype safe and happy and, just well looked after. Paratypes are going to be older or younger than the holotype, so they'll show different stages of development. Paratypes may show the insides better. You may have an individual that has kind of has all of its little horns and bumps and spines intact. But it's not as complete as the holotype. You may only have, part of it. That's a great one to be a paratype. Yeah, because you wouldn't want it to be the holotype, because then you would be comparing it to a specimen with parts missing. And then what do you fill in for the parts that are missing? That's tough. Yeah. So the paratypes support the type, but the paratypes do not define the type. They're just helper types. Yeah. Because the holotype is the one and only type. Now, if you're doing things right, the paratypes should be from the same location, same time, same place as the holotype. Because you want to make sure they're the same species, because species change with distance and with time. Yeah. So it's hard to be sure that individuals are really, truly members of the same species, especially when you can only see certain parts of each one. Like take a brachiopod shell. If it's closed, what does it look like on the inside? You don't know. Nobody knows. But if there's only one kind of brachiopod at the site, and you've got a whole bunch of them, some of which are open, some of which are shut, some of which are just half or part, then you can build up a picture of what this thing is. So that's what paratypes are for. Yeah. Syntypes. This is these are a sin, basically. Yeah, SIN. Syntypes are from back when you used to designate a group of specimens to become the type specimen. It was like a type club, and you would put several things inside of it. Problem is, with a lot of those early syntype groups, they're all from the same place, but somebody will put like all the corals from that place into a pile and say, yep, you're a bunch of syntypes. And there may be 3-4 different genera in there. There may be multiple species within those genera, and it may just be a mess. That's why we have a holotype, so it's at least just one. You know that an individual can only be part of one species, so at least you don't have that problem. So when you encounter syntypes in the world, it is your job to select and then publish that selection. One of those, usually the biggest and oldest one, like it says up there, and that will become the type. And so, yeah, weirdly, we have half the rules for types up here. Yeah, I don't know. That was not what we intended to have. So the selected one becomes the lectotype because you select a lectotype. Yeah. What happens if your type is in a museum in Germany or Britain, which is bombed all to heck in World War II, and the museum is reduced to ash, and so is the fossil, and so it's gone? In that case, you have to select a new type, and often that means going out and recollecting, hopefully at the same site if the records are good enough from the publication, because the whole point of publishing is to distribute the information so that if something bad happens to the holotype, then something bad doesn't happen to the whole species. Because if you can't tell what a species is supposed to be, then that species is invalid. And taxonomists all the time have to do this, and it hurts because you have a name that people have been using, or at least could have been using, maybe for hundreds of years. And if you can't tell exactly what it is, and you can't clean it up by designating either a lectotype or a neotype, depending what's available, then you just have to blow up the species. The species no longer exists. The specimens exist. You have to either make a new species to put those things in, or attach them to a name of some other species that's already out there. Those are changes you want to take very carefully because they are disrupting the current taxonomic structure, and so you don't want to do that. That is your job, to be very conservative with species names. All right. And then there are plenty of other types, like plastotypes are one of my favorite. That's if you have a gigantic rock, like massive boulder in the field that has a fossil on it. You can take a plaster cast of it, or you can take a little rubber cast or silicone cast or something like that. And if it's a plaster cast, you call it a plastotype. Or if you take a little rubber cast and then you make copies of the fossil, basically, but in plaster, then those are plastotypes. Yeah, that's the word. And there's a bunch of others. Yeah, but we don't need to know the others. These are the common ones. Holotypes and paratypes, those are good types, the real one and supporting ones. Syntypes are bad types, so you got to select one, a lectotype. And neotypes are somebody coming along, bless them, and trying to patch over a horrible hole in the taxonomy where there's a species name, but there is no type specimen anymore because something bad happened. Yeah. All right. So how do you designate these species? Where do you draw the line? That is a thing that differs from worker to worker. Now, I can tell you that a good species description is going to include things like sexual dimorphism, if that appears to be something that exists. It is going to include development. So if there's a big metamorphosis that you can see, then you need to record it. You're like, oh, okay, it looks like this early, and then it changes into this. A species can have at least some mathematics in the description. You can say it's like 3 times as long as it is wide or something like that. Ballpark. And treat those numbers as kind of squishy. but those are a way of separating one species from another. If you have one that's really long and one that's not so long, but they look fairly similar, and there's no way that this could have grown into this, then you can designate it that way. Often, the species concept is going to involve mathematics. And so, like here, we're taking the same brachiopod at different ages, and you can either read out the little growth lines on here to see how wide and long it was at different times, because everywhere that it stops, it's going to stop all along the shell. And so you can see those grow, and look at those make a really nice linear relationship. Or you can take a bunch of individuals of different sizes, from itty-bitty babies to great big ones. And each of those will define a line. The lines may be distinctive, like a growth ratio is sometimes helpful in telling one species from another. But if you're across several genera or within a family, you'll have several of these things that pretty much line up on top of each other, because there's a shared set of genes that are determining how it grows. That's just how it is. All right, names. Once you have your type specimen, and once you have a description, that's the set of rules that tell you, all right, who gets in, then the next thing that you need is a name. And the species name itself, that designation you get to pick, because a species is a group of individuals based on one key individual, a type individual. And then you also designate a genus. And so if it's part of a genus that's already been named, that's fine. You just say genus with a name, and you give it a new species name, and you define it, and you say, here's the description, here is the type specimen, it's in this museum, go look at it if you want. You show pictures of it now in the modern age, because we don't have to sketch things or make a word picture. Yeah, because a real picture is much better when you can get a nice clear one anyway. And then for higher taxa, this is the neat part. Higher taxa will be automatically generated from your genus. So if you make a new genus, like say you make the genus Rugosoconides, that will generate on top of it a subfamily and a family automatically. And this goes up to family level. I'm not sure if it goes to super family. I think it just goes to family level, but I don't know. We could argue about that. And each of these levels has a particular ending. And so if it's Rugosoconites, ES, then the subfamily is going to be an INE, and the family is going to be an IDE. And so here's Conites, Conetinae, Conetidae, Productus, Productinae, Productidae. Yeah, there's a particular ending that should define and should tell you. If I just throw out a term, Rugosoconidae, you'd be like, oh, it ends in ide, so it has to be a family. Yeah, you know it from the name, which is nice. It's nice when science is something you can figure out, not something that you have to memorize. So you memorize a couple of name endings, and then you can recognize the rank of anything. Now, of course, in the old days, before they standardized things, the bad old days, people used to make names of species and families and whatever, and they didn't always follow all the rules. These came into practice over time. So most species, genera up to families, will follow these rules. People have been going back and cleaning up these wherever they can. When we are above family groups, these are things that are not automatically generated, but they are designated, and they're usually designated with reference to a particular genus and species. So each genus has a type species. Each species has a type specimen. So the type species is based on a type specimen, and the type species determines the genus. So the genus, you would say, okay, Rugosoconides, this particular name, Celticus, is based on this particular species, Rugosoconides celticus. But the whole genus Rugosoconides is this species and all the ones that cluster around it. That's what a genus is. It's a collection of species. A subfamily is a collection of genuses, and in science we say genera is the plural for genuses usually. And a family is a group of subfamilies, which contain genera, which contains species. Yeah. At its simplest, there's only one species in a genus, one genus in a subfamily, one subfamily in a family. And that just means you probably need to get out there and look for more fossils, because you've got a lot of missing branches on your tree. You got one wiggly stick, and you're kind of missing all the side bits. Okay, now what happens if a species that's been used to build all of these automatically generated or deliberately generated names above it? What happens if you take that thing and then you you figure out that, wow, there's no type specimen. The original description is not very good. The site where it came from is under blacktop and houses now, so there's like no way to go back and get another one. Because if you knock on somebody's door and say, I need to jackhammer a hole in your floor because there might be a little tiny fossil down there, and we've got a name for it, but we need one that we can hold on to and put in the museum, I think they're going to say no 99 point, a very high number of the time. Yeah. So when that happens, the species actually becomes invalid because you can't substantiate it. There's not a good enough description for it. Maybe it was only a partial specimen that was described, and maybe that specimen has been lost, and maybe there's no way to go back to the site because it's gone, so you can't designate a good neotype, and you're not really sure what you should even designate as a neotype. In that case, yeah, the name of that, the names attached to that, the species and genus and any of those automatically generated names above it become invalid. And once they're invalid by the rules, the ICZN over here, they make the rules, yeah, that becomes unusable. So you cannot use a name which has been invalidated for any reason ever. And that is just to keep sanity for taxonomy. It is just to make it so that you don't have the old names and new names competing with each other. So this is why you have big fights, like when the famous trilobite Phacops turned out to be actually named Eldridgeops because somebody published a partial, and later people figured out, these are the same thing. Whichever one gets published first, there's a rule of priority. So the earliest one to be published that accurately describes this species is the one that gets to carry the name, and that's the name you have to use, whether you like it or not, going forward. Yeah, it is difficult to get around that. But when you're dealing with these names, remember that each of these names actually comes from something. So there's a genus, let's say it's Lysoconides right here. And that genus only exists because of one species. There are maybe 5 or 6 species in that genus, but only one of them is defining the genus. And if that one gets invalidated, then yeah, you have to find a new home for all the others. So here is Lysoconides, and these are 2A might actually be one of the types that was originally designated from a set of syn types for this species, and that's what the genus is built on. And then that genus sits in, this is the full taxonomy for one brachiopod, the Svalbardinae. Svalbardinae is a subfamily that's defined based on Svalbardia. Here's Svalbardia in red. That sits inside the family Rugosoconidae, which is based on Rugosoconides. So if Rugosoconides turns out to be invalid, then that blows up Rugosoconidae, Rugosoconidae, and Rugosoconides, the genus, and we have to find new homes for a whole bunch of fossils. We try not to do this. You try to build a taxonomy that is not going to fall apart, because if your taxonomy falls apart, you have not been doing your job as a taxonomist. I mean, come on, yeah. So anyway, Rugosa sits within the superfamily Conitidina, which is based on Canides. Now, this is above family level, so this one can continue to exist even if the genus that it's based on and the species turns out to be part of a different genus that already existed, that had priority. And there are actually a lot of these names in the literature at superfamily, suborder, names of things that used to be like the number one, the most common version of this particular type of organism, but has since had its name changed because the name, it belonged in a different group or there was some problem with it. And so, yeah, you'll have these little ghost taxa above the family level. And anyway, that's part of, so that one defines the superfamily and suborder. The order is defined by Productida. Here is Productus, the leader of the Productida. It belongs in class Strophominata. Here's a Strophomina, which is just a genus, just a humble genus. But this is a genus that has carried a subfamily, family, superfamily, suborder, order. There's a Strophominida, order, and a class, which is the Strophominata. So all the things that are similar to Strophomina in some particular aspect belong in that class. And those belong inside the Rinconelliforma, which is named after Rinconella. And here is the original Rinconella at the bottom. So that's kind of cool. So when you're saying the taxonomy, you're saying, okay, it's one of these. It's got these features, but it's this particular type of those. It's got a flat hinge. Like, oh, okay, flat hinge, not a weird, crazy curved hinge. Got it. And you're like, yeah, flat hinge, but it's got a flat hinge and it's got spines coming out the back. You're like, whoa, spines, what? Yeah. And then you come up here and say, yeah, it's got all that, but it's kind of flat and shaped like the letter D turned over on its side. And like, okay, but all those other things are true. Yeah, still true. And then you say, yeah, all right. And it's one of those, but this is going to be one of the ones that instead of being like variable on the shell, it's going to have these little ridges that run all the way down. It's going to be rough. Rugose. That's what rugose means. It means rough. And it's usually going to mean it's got lines running down it. You're like, okay, fine. It's going to be part of this big group of things that have lines running down it. It's like, yeah, but there's a couple of members that are related to those, but they don't have the lines. And so, all right, those are the Svalbardinae. Fine, I got you. And then within the Svalbardinae, you can say, well, yeah, there's Svalbardia, and that looks cool there. Look at this massive median septum that goes about halfway down and great big little arcing things. But what if you have a median septum that goes all the way down, or some major difference inside? Then you can say, well, all right, it can't be in Svalbardia. We have to make a new genus inside there, and so we'll call this Lysocanides, but this is its taxonomy. There you go. That's how it works. This particular example, not important. Yeah, it's just one that I happen to know. But the idea, I want to make sure you got the concept for how these things are stacked up and how they are built, and how everything family and below can be destabilized if one specimen, which defines a species, gets lost or damaged or can't be, we don't know what it is anymore, or wasn't published with a good photo. Yeah, that's just how it goes in taxonomy. All right, so study questions. What's a species? Group of individuals defined by rules related to a type specimen of some sort. With a taxonomy, which includes a name. That's basically the fundamental elements for how a species exists. It's just a set of rules, which are how we're going to divide up biology. On a functional level, like, when you're actually doing stuff, if it's closer to members of the species than it is to any other species, then you'd say, yep, it's within the species. But there are edge cases where you're trying to draw, like, what's the boundary of this species, and those can be tough. What's an operational taxonomic unit? So this is something that's been designated. So something like a species, a genus, a subspecies. It's just a group of shells. And so you can have operational taxonomic units that fall in between genus and species, like subgenus, but you can have as many levels as you need. They're not going to be official taxonomic levels, but any way that you can group more than one individual together, or more than one, species group of individuals together, that's going to be basically a way to do it. And so we call this an OTU, Operational Taxonomic Unit. If you think of a phylogenetic tree, the branches, each little tip, that is usually going to be an individual. But everywhere that the tips join, those are going to be OTUs. And so all the points where the family tree branches, like all the ancestors, are operational taxonomic units. You can do this with your family. Yeah. Your kids are going to be descended from you. You're going to be like an operational taxonomic unit over your kids. And your parents and their parents and their parents and their parents, all the little dots on the tree, all the way back as far as you can get and as complete as you can be. Those are all OTUs. Yeah. What's a type specimen? What's its purpose? There's a whole slide about that. Why don't you go look at it? Yeah. Different kinds of type specimens. Oh my gosh. Yeah. What's a paratype? What's a syntype? Oh, it's a syn. That's for sure. Yeah. Go back and look. Write up a little thing on it. Then you'll have it. You'll have the idea. How do we objectively separate species, especially when fossil DNA is gone? That's tough. We've got a whole other lecture coming on this, but you're going to base it on the description, and you're going to, different features are called characters. So you'd say, what's a particular character, like a nose that looks like this, something like that. And you'll define that in words, possibly mathematics, and that will be a character. And then a set of characters will make up the description. Like, to be in this group, it should look like this. You describe each part. Usually you'll do it in a particular order, so it's not just chaos. You go from head to toe, go from posterior to anterior, whatever. Yeah, whatever makes sense for the group. So that's how we do our best. And you can make family trees from these character matrices that are basically just as valid as DNA family trees. All right, what does it mean to say that the genus is the standard operational taxonomic level? So in paleontology, the genus is kind of the most important. It's going to be based on a species, but the genus is the one that makes the subfamily and family, and it also makes a subgenus as well. And so those are all going to be like the children of the genus. And so when you read fossil books, generally they're going to have the genus in there. That's going to be the important thing. And it doesn't really matter which species you mention, because that species only gets to exist in relation to the type species for that genus. So often when you have a genus, they'll have like Conotanella ramsbottom, and ramsbottom is the one who defined what Conotanella is and posted the pictures of the species that became the genus type, which is also called a genotype. Yeah, which is weird because they use that word also for like genetic type. And so we're not supposed to use that word anymore, but yeah, you will see that in older literature. And let's say you have a family. The family is based on a genus, some genus within that family, and there's probably an auto-generated name. This happens automatically. You don't have to do anything. The names are just generated once you have a valid genus. And you generate enough names until you have joined on to some point on the tree. So you only generate as many names as you need, and the rest of the names that could have been still can exist if you find out your old taxonomy was wrong, but usually, they just go away. They never get to exist. All right, and we talked about taxonomic collapse, which is basically the second half of that question. So I think you've got a lot of the tools that you need now to understand taxonomy. And I don't know, I think that makes museum trips a little more interesting. Often you'll see on a specimen like a little dot, usually a little colored dot. That is usually how museums mark the holotype or one of the paratypes of a species. And so if you're in the right museum looking at the right display, you may be looking at the Tyrannosaurus Rex, because there is only one, and all the rest exist only by comparison. So that's kind of neat, And sometimes it'll say it on the little description card. So it's why you got to read those museum cards if you're going to the museum. All right. Hope that helped. Thanks very much.